Good morning. Today I would like to give lecture on immunoglobulins. Immunoglobulins are nothing but these are protein molecules. They belongs to the glycoproteins. They are involved in the protection of host or humans from different types of pathogens. So, they eliminates the pathogens, they interact with the pathogens with the mechanisms like pathogens. They do have different types of proteins and lipid molecules. So, these molecules when they enter into the body, they act as a antigens. So, they stimulates the immune response. Due to stimulation of that immune response, they produce the immunological molecules. One of them is immunoglobulins. So, these immunoglobulins interact with that antigens and they neutralize the antigen, or they eliminates the antigen, or that antigen containing pathogen will undergoes into the phagocytosis mechanism, or it undergoes into the opsonization, or it will be the toxin of the pathogen will be neutralized. So, these are some of the reactions or uses about the immunoglobulins. So, today I would like to focus on the basic structure of the immunoglobulin and the what are the functions about the immunoglobulins and what are the classes about the immunoglobulins. So, basics about this immunoglobulin topic. So, basically in our body we do have the immunological cells, example called T cells and B cells. So, they interact with the antigen with the help of antigen presenting cells. After that they will be converted into effector cells. So, these effector cells they produces the memory cells or memory cells of the B cells, then they convert into plasma, they produce the antibodies. So, these antibodies we are calling as immunoglobulins. So, this pathway also called humoral immunity. Another pathway that is known as cell mediated immunity. In the cell mediated immunity, so the T cells, the T cytotoxic cells directly interact with the antigen with the help of antigen presenting cells. They produce with the various types of interleukins. Due to production of that interleukins, they kills the cells. This mechanism or this immunity known as cell mediated immunity. So, our antibody production occurs only in the humoral immunity mechanism. So, other some of the words like antigen, what is the meaning of antigen? How you will define it? Any substance that can binds to specifically its producing product, that is known as antibody. So, this antigen has epitopes. So, these epitopes interact with the antibody paratopes. Another word that is called immunogen. What is the immunogen? Immunogen is nothing but any substance that can induce a specific immune response inside the host. So, that immune response created by this antigen, that antigen known as immunogen. Another one is epitope, the antigenic determinant which is present on the surface of the pathogen or which is present on the surface of the antigen, or the part which can interact with the paratope of the antibody, that is known as epitope. Another one is aptane. So, this aptane is a antigenic molecule, but it do not have the immunogenic activity. As such, this aptane do not stimulates the immune response. It requires a carrier molecule. With the help of carrier molecule only it produces the antibodies. Another one is paratope. So, what is the meaning of paratope? Paratope is nothing but a antigen binding site which is present on the immunoglobulin or which is present on the antibody. So, this paratope in turn it interacts with the epitope of the antigen, then it will form the some of the non ionic or non covalent interactions. Due to that antigen will be eliminated. Coming to the structure of immunoglobulins. So, these two scientists they worked on this invention of the antibodies. One name is Tiselius and followed by the Cabot. So, they have taken the ovalbumin, this ovalbumin injected into the rabbit, Then after injection of the rabbit, after 3 to 4 days, they isolated the serum of that rabbit, then they subjected into the electrophoresis, then followed by the gel filtration chromatography. In that they got these kind of peaks. So, one of them is ovalbumin, then followed by the alpha peak, then followed by the beta peak, and then followed by the gamma peak. Then further to these fractions again they added the ovalbumin. So, this ovalbumin specifically, it removed the gamma fraction, it removed the gamma protein from that fractions. So, then they isolated, then they given the name as IgG or immunoglobulin G. Later on Gerald M. Edelman and followed by the Porter, so they worked on more on IgG molecule, then they invented the structure of the antibody or structure of the immunoglobulin G molecule. So, for that they got the Nobel Prize in 1972. So, by contribution of these scientists, so we came to know that the structure of antibody so looks like this. So, this antibody basically it has two regions, one is known as, it is basically a Y shaped molecule. So, this Y shaped molecule it has two basic regions, one is called antigen binding site or the paratope, another one is called FC portion or fragment of crystallization. So, these are the two basic parts which are present inside the immunoglobulin. So, this FAB they also called as FAB and FC portion. FAB portions involved in the binding to the antigen or binding to the pathogen, whereas this FC portion involved in the other effector functions like complement activation, opsonization and neutralization or receptor binding processes. So, this is the basic structure of the immunoglobulin. It has FAB fragment of antigen binding site, another one is FC fragment of crystallization. Later on because of action of different types of enzymes and different types of chemicals, then they invented this structure. This structure of the immunoglobulin, it has two identical chains. So, one chain is called light chain, another chain is called heavy chain. So, in the light chain it has again the light chain divided into two regions, one region is called variable region, another region is called constant region. Same way in the heavy chain also you will find two regions, one is called variable region, another one is called constant region. So, I would like to draw on board to clarify this. Okay. So, this is the basic structure of the immunoglobulin. So, here this is called light chain, this is called heavy chain, okay. So, this light chain again it belongs to the proteins, that is why I given the name as, I told name as immunoglobulin or globular protein. So, it will start with the first amino acid, it will start with the N terminal and it ends with the carboxylic terminal. Same way in the heavy chain also it start with the amino terminal and it ends with the carboxy terminal. So, this light chain it has two regions, one region they used to call variable region of light chain, constant region of light chain. Same way here also this is also light chain, it has two identical light chains, variable region of the light chain and constant region of the light chain. Same way inside the heavy chain you will find different regions. One of them is variable region of heavy chain and variable region of constant region of heavy chain, constant region of heavy chain. So, like that way inside the heavy chain you will find, instead of one you will find different constant regions, they given like constant region 1, constant region 2, constant region 3. Same way here also variable region of heavy chain, constant region of heavy chain CH1, CH2, CH3. Then this part and this part involved in the it binds to the epitope of the antigen. So, that is why this part they used to call paratope. Then inside the light chain you will find the some of the domains. So, these domains will form the intra disulfide bonds, Same way also in the constant region also you will find the intra disulfide bonds. So, this light chain has 210 amino acids length, 210 amino acids in length, and it has two types based on the constant region of the heavy chain. They are lambda type and kappa type. So, based on constant region of the light chain divided into two types, one is called lambda chain, another one is called kappa chain. Then this light chain it can also form the interdisulfide bonds with the heavy chain. So, because of the formation of the interdisulfide bonds it joins to the heavy chain. Next one is heavy chain. So, in the heavy chain variable domain will be there, then followed by the constant regions like CH1, CH2, CH3. Here also in each domain you will find one intradisulfide bond. So, this intradisulfide bond it occupies 70 amino acids, 70 amino acids. So, constant region because of the length and followed by the because of presence of amino acid sequence, this constant region of the heavy chain again divided into different types, like they used to call alpha, beta, gamma, epsilon and delta. So, these are the 5 types. So, based on this constant region of the heavy chain amino acid difference, these immunoglobulins or these antibodies are divided into 5 types. That is why they used to call GAMDE. So, GAMDE like immunoglobulin G, immunoglobulin A, immunoglobulin M, immunoglobulin D, immunoglobulin E. So, where we get from these different types of immunoglobulins based on the constant region of the heavy chain, based on the constant region of the heavy chain amino acid difference, that immunoglobulins are divided into 5 types. So, this portion known as FC and this complete portion known as FAB, fragment of antibody binding site and fragment of crystallization. So, this fragment of crystallization involved in the different types of effector functions inside the cells. Okay. So, these are the some of the intradisulfide bonds, then followed by the interdisulfide bonds. So, this is the antigen binding site and this is the FC portion, and between the light chain and heavy chain you will also find the interdisulfide bonds, and inside the heavy chain you will also see some of the attachment of carbohydrate moiety. So, this presence of the carbohydrates basically they bind to the constant heavy chain of the constant domain of the heavy chain 2. So, because of binding of these carbohydrates, so that is why we used to call as glycoproteins. These immunoglobulins called as glycoproteins. So, this presence of carbohydrates, they can also involved in the complement activation. So, these are the some of the domains. So, basically in the IgM and IgE, you never see one special region, the region known as hinge region. So, this hinge region absent, whereas when you go into the G, A and E, so there you will see this hinge region involved in the movement of the antibody or movement of the immunoglobulin, like they get the flexibility because of presence of the hinge region, that immunoglobulin will get the flexibility. So, the flexibility possible by only presence of one special amino acid, that amino acid known as proline. So, because of the presence of the proline, it moves different directions. So, next one, another one is the special important one is known as antigen binding site. So, this antigen binding site are the variable domain of the light chain and as well as variable domain of the heavy chain. So, these two chains, they will form the variable domain. Inside the variable domain, some portion of the antibody only binds to the antigen or epitope of the antigen. So, these regions, they are known as variable region. So, inside the variable region you will find the hot spots or they can also call as complementary determining regions. So, these complementary determining regions, in the light chain you will find 3 hot spots, whereas in the heavy chain you will find the 4 hot spots. So, these regions only they interact with the epitope or antigen part. So, this is the immunoglobulin structure, this is the antigen which binds to the paratope or the variable domain of the heavy chain and as well as light chain. So, like that way it will form the groove. So, the inside the groove they binds to the that groove with the help of non-covalent interactions. Next one is chemical reactions of these immunoglobulins. When you add different types of enzymes to that immunoglobulins, they will show different types of they will be converted into different types of polypeptides. When you treated with the papain, so that will be cleaved into different portions, like you will get the single portion of the FAB and FAB, then followed by the FC portion. Whereas, when you treated with the pepsin specifically cleaves the FC portion of the antibody. Due to that, you will get the 2 FAB means the FAB portion linked with disulfide bond. Whereas, when you treated with the different types of chemicals like mercaptoethanol, so this mercaptoethanol specifically it cleaves the or it reduces the disulfide bonds, disulfide bonds which are present between the heavy chain and as well as between the light chain and heavy chain. Due to that, you will get the 2 bands, one is called 25 kilodalton band, another one is called 50 kilodalton band. The 25 kilodalton band known as light chain, 2 identical light chain, then followed by the 50 kilodalton band known as heavy chain. So, next one is coming to the immunoglobulins, types of immunoglobulins. So, based on constant region of the heavy chain, difference in the amino acid sequence of the constant region of the heavy chain, immunoglobulins are 5 types. They used to call GAMDE, immunoglobulin G, immunoglobulin A, immunoglobulin M, immunoglobulin E and immunoglobulin D. So, this is the general overview of all immunoglobulins. So, this is the class like G, M, A, E, D, this is a heavy chain. So, like the constant region of the heavy chain, they belongs to the gamma type, that is why they are given as IgG. The heavy chain belongs to the constant region of the heavy chain belongs to the mu, so that is why they given the M, and the heavy chain belongs to the alpha type, they given the IgA, and the heavy chain belongs to the epsilon, so then given the IgE, and as well as the heavy chain belongs to the delta, they given the IgD. So, apart from that, these immunoglobulins they tend to have the subclasses. So, when you see in the IgG, you will find the 4 subclasses, again difference in the constant region of the IgG molecule. So, they are gamma 1, gamma 2, gamma 3, gamma 4 or G1, G2, G3, G4. Then in the M you do not find any classes, subclasses. Then in the IgA you will find alpha 1, alpha 2 or Ig A1 and A2. In the E and in the D you never find any subclasses. Then the coming to the light chain of these immunoglobulins. Basically in the light chain of the humans, basically in the humans you will see 40% of the kappa and 60% of the lambda. So, any one of the will be there, any one of the light chain will be there, but you will never see both. So, based on this you can write the molecular formula like 2 heavy chains and 2 light chains, 2 heavy chains and 2 light chains. So, in the G you will find G always exists as a monomer, but whereas when you go into the IgM it will exist as a monomer, dimer and tetramer and pentamer and hexamer. So, like that way you can write the molecular formula of these immunoglobulins like N is equal to 1 or 2 or 3 or 4 or 5 like that. IgJ it will exist as 2 types like dimer and as well as monomer, and IgE and IgD they always exist as a monomers. Sometimes this IgD it is present on the surface of the B cell. So, that is why this IgD also involved in the antigen presentation. So, the first one is properties about the immunoglobulin G molecule, immunoglobulin G or IgG or antibody G. So, basically this antibody it forms inside the serum. So, basically it is present inside the serum, sometimes it is also present in the tissues, but most of the serum the total antibodies this occupies the major portion like 70 to 75%. So, they basically it is exists as a monomer, it has the molecular weight about 1,50,000 Daltons. It can cross the placenta. So, that is why they used to call maternal antibody. So, because during embryonic stage to protect the fetus, this IgG transfers from mother to the fetus with the help of umbilical cord. So, that is why that IgG called maternal antibody. So, it is also called warm antibody, basically it requires 37 degrees Celsius for the reaction purpose, and also it is produced during the secondary immune response. When you see the immune response, basically it is 2 types, the primary immune response and the secondary immune response. In the primary immune response, the initial infection, during that initial infection only some of the antibodies are produced, one of them is IgM will be produced, whereas after later infections after 21 days, after 30 days or in the secondary infection or in the secondary immune response IgG is produced. So, that is why they used to call basically this IgG produced only during the secondary immune response, and it has the half life about 21 to 23 days inside the human body, like in the serum it will find up to 21 to 23 days. So, that is why the initial infection, the primary infection, so they will look for the IgG molecule by ELISA or some of the serological diagnostics. After 21 days or after 30 days they will go for the IgG only, before that they will go for the IgM, and also it is exists as a monomer. It contains 2 to 4% of the carbohydrate percentage, and also called it is a incomplete antibody. So, this incomplete antibody basically it is formed for the Rh factor which is present on the surface of the RBC. So, that is why they also called anti-Rh factor. So, this IgG can activate the complement, and it can function as a opsonin, and it can also react with the heterophile antigen. So, this is the picture related to the transport of IgG from mother blood to the fetus with the help of placenta. Basically, the placenta cells possess the receptor for the IgG. So, that is why it will be traversed from mother blood to the fetus. So, that is why it is giving passive immunity towards that baby. So, these are the subclasses about the IgG like G1, G2, G3 and G4. So, next antibody is that IgA. So, this IgA it is present in the serum, it is low concentration, whereas in the fluids it is present in more concentration. In the body fluids or in the secretions it will be present as dimer. So, it has 10 to 15% about the percentage in the serum, and also it has 8% about the carbohydrate moiety. It has two subclasses, one is called A1 IgG A1 and IgA2. So, A1 is present in the serum, so whereas A2 present in the fluids or secretions like colostrum or sweat, sebum or urinotract, genitourinary fluids. It will basically this A2 IgA2 basically it is present in the fluid, or it will give the mucosal immunity towards the various types of pathogens. It has half life about 6 days. So, these dimeric IgG molecules or dimeric IgA immunoglobulins are joined by with help of a small peptide, that peptide known as J chain. It has 15,000 Dalton molecular weight. So, this 15,000 Dalton molecular weight J chain linked with the Fc fusion. So, this J chain has more amount of cysteine rich amino acids. So, this because of presence in the breast milk. So, this breast milk passed to the baby, newborn baby. So, that is why that newborn baby will get the passive immunity from the mother with the presence of this Ig immunoglobulin with specifically IgA2. Next immunoglobulin is that IgM. So, this IgM it is the highest molecular weight among all immunoglobulins. It can exist as all different types of monomer and as well as pentamer and as well as hexamer. So, monomer basically it is present on the surface of the B cell. So, this monomer IgM involved in the interaction with the antigens, that is why this IgM involved in the antigen presenting interactions, and pentamer and hexamer present in the serum, and they are involved in the blood-borne elimination of the blood-borne pathogens. They are also linked with J chain, and it has the sedimentation coefficient about 19 S, and in the serum you will find the 10 to 50%. It has the carbohydrate moiety about 12%, it has the half life about the 5 days. So, this antibody produced during the primary immune response. So, that is very very important during initial infection. So, IgM will come, whereas later infections like in the secondary immune response IgG will come. It cannot cross the placenta, and it is also called or it is produced by the neonate and or fetus. So, that is why it is the first producing antibody inside the body. The first producing antibody in the humans or the first producing body inside the newborn baby is IgM, that is very very important. So, it is also involved in the agglutination process. So, basically this IgM reacts with the particulate antigen. So, that process known as agglutination. So, that is why this antibody also called agglutinin, and also known as cold antibody or complete antibody. So, because the reaction will be more in the cold temperature, and when compared with all the antibodies, specifically IgG. So, this IgM has highest complement activity, and also it is produced for the rheumatoid arthritis or autoimmune disorder. So, that is why they also called rheumatoid factor, and also called Wassermann antibody. So, this antibody is produced for the Salmonella antigen, Salmonella O antigen, nothing but type E, Salmonella type E and Salmonella paratype E. Next one is IgD. So, this IgD which is present in low concentration inside the serum. So, basically it will exist or it will be present on the surface of the B cell. So, this B cell, because of presence of the IgD, it is involved in the recognition of the antigens inside the blood, and also it is involved in the presentation of the antigen. And also this IgD, it is produced during the some of the autoimmune disorders like thyroid disorder and insulin dependent diabetes, and also it shows the reaction with the penicillin and as well as diphtheria toxoid. So, next one is IgE. So, this IgE is also present in the low concentration in the serum. It has 1,90,000 molecular weight. It has the sedimentation coefficient tables 8s. It has the shortest half life like 2 to 3 days, and carbohydrate moiety is 12%. It is always exists as a monomer, also called PK antibody or neogenic antibody. So, because of presence of this antibody inside the serum, it leads to some of the hypersensitive reactions. So, basically the allergic reactions are produced by presence of IgE. So, this IgE reacts with the allergens. So, this IgE as receptor on the surface of the mast cells. So, mast cells are basically the type of cells which are present on the inside the body. Basically they belongs to the basophils. So, because of presence of this IgG, it binds to the its receptor on the mast cells. Due to that, degranulation of that mast cells leads to production of histamines and serotonins. So, these two molecules are involved in the different types of allergic reactions. So, these substances, these are involved in the different types of allergic reactions. So, conclusion. So, in the conclusion, so immunoglobulins are nothing but glycoprotein molecules. So, they are involved in the various biological functions, and they are different types. They made up of 2 different types of heavy chains and 2 different types of light chains. They are linked with the disulfide bonds, and they have the 2 major portions. One is called fragment of antigen binding site, another one is called fragment of crystallization. So, this fragment of crystallization involved in the different types of effector functions like activation of other cells and activation of complement or activation of opsonization or activation of phagocytosis. So, like that way, whereas fragment of antigen binding site, it specifically interact with the antigen and it will form the different types of non-covalent interactions. Due to that, the antigen will be eliminated with the process of neutralization or agglutination or precipitation reactions. Then based on difference in the heavy chain, immunoglobulins are 5 types. So, most prominent one are G, A, M. So, G always present inside the serum, and as well as sometimes it also present inside the fluids. So, this G is produced from the secondary immune response. IgM also present in the serum. So, this IgM involved in the primary infections, it is produced during the primary infections. So, there is a basic difference between the IgG and IgM. IgG has more avidity, means it can bind to the more than one antigen, whereas IgG has low, it has more affinity. So, these are the 2 different words, one is avidity, another one is called affinity. So, affinity has more towards the IgG, avidity has more towards the IgM. So, whereas IgJ IgA always present inside the fluids. So, all the body secretions, colostrum or breast milk, so skin, sweat and sebum, like that. And D always present on the surface of the B cell. E, it is present in the serum, but it is involved in the some of the adverse allergic reactions. So, this is about the structure of the immunoglobulin and as well as functions about the immunoglobulins and different types of immunoglobulins. So, these are the two reference books. You can follow the QB Immunology and Immunology by Wright and Wright. So, these are the two standard reference books. So, that can be useful for the various types of immunological process. So, you will find all the matter related to the paratope or epitope, immunogen, different kind of small mark questions you will find in this book, and as well as this is the very important question in the long essay question. So, definitely you will get in your annual exams. So, just like they will ask like describe the structure of the immunoglobulin or describe the various types of immunoglobulins. Thank you very much. హలో
హలో సార్
చెప్పండి సార్.
సార్ హలో
చెప్పండి మేడం.
సార్ నేను హలో సార్ నేను నరేష్ మాట్లాడుతున్నా సార్.
చెప్పండి.
ఐజీజీఏ ప్లాజెండాను చేస్తుంది, మరి మిగతా ఎందుకు చేయాలి?
మళ్ళీ ఒకసారి చెప్పండి
సార్. ఐజీజీఏ ప్లాజెండా చేస్తాను, రియాక్షన్ చేస్తాను కదా సార్. ఐజీఎం ఎందుకు చేయాలి?
ఓకే. సో ఎందుకంటే సో బేసికల్గా ఐజీజీకి ప్లాసెంటాలో ఉన్నటువంటి సెల్స్ పైన రిసెప్టార్ ఉంటుంది. ఐజీజీ రిసెప్టార్ ఫర్ ది ఐజీజీ. సో బికాస్ ఆఫ్ ప్రజెన్స్ ఆఫ్ దట్ రిసెప్టార్, సో దిస్ ఐజీజీ ఫ్రమ్ ద మదర్ బ్లడ్ it goes and binds to the receptor present on the placental cell. So, that is why it will form the one of the cyst inside the placental cell, then it will be transported to the baby blood. So, that is why it will traverse from one mother blood to the baby blood, whereas when you see IgA do not have the receptor. So, that is why it cannot be transported. So, in the IgG specifically G1, G3, G4 all will be transported, whereas G2 cannot be transported because G2 also it lacks the receptor on the placental cells. So, that is the answer. Thank you.